that isn't able to join. Got it. I'm trying to tap on my screen of my laptop because I'm so used to everything else being touchscreen. All right, welcome everybody. We're going to go and start with our guest speaker. We have Dr. Jon Kim with us tonight. Welcome. He's a professor at the UF Veterinary School, correct, Dr. Kim?
Yes, correct.
And doing some research with hemangiosarcoma. So if you want to go ahead and start off, I'll turn it over to you.
Okay, can everyone see my screen?
Yes.
Yes. Okay, sounds good. All right. Let's start. And all right. So hello everyone. Thank you very much for joining, and thank you very much inviting me to this meeting today. My name is Jon Kim. I'm an assistant professor at the University of Florida Vet School, and I'm a veterinarian and a PhD researcher focusing on the canine cancers. I'm using cutting edge technologies such as AI and machine learning. And I'm really very excited and honored to be here and to present to you. My entire career has been dedicated to helping sick animals, especially for dogs with cancer. And in particular, I have been doing research on golden retrievers. So I'm truly excited, and words cannot fully express my excitement about this opportunity. For this talk today, I'm going to discuss new origins and hope for hemangiosarcoma in dogs by briefly sharing what we have been doing and what we are currently doing in research for this disease. So today, the presentation outline includes an introduction and history of hemangiosarcoma research, including our previous work, and a focus on our current research efforts. First of all, I'd like to introduce myself by focusing on my academic journey in canine cancer research, because I'd like to share my philosophy and perspectives on my work and my job. I was born and raised in Seoul, South Korea, which is very far away from here, and which is across the Pacific Ocean from here, about 6,250 flying miles away. And I completed my academic degree in veterinary medicine and obtained my PhD in veterinary pathology here. And originally, my career goal was to become a veterinary clinician to help actual sick animals in clinics. However, during my studies in veterinary school, I realized that there were still a lot of things that we could not do, especially for animals with devastating disease such as cancers. So that's why I pursued my PhD work in pathology, which gave me the opportunity to advance knowledge about disease diagnostics and fundamental pathology. However, when I did graduate work in South Korea, research for dog cancer was very uncommon. So I was not able to do advanced research work for focusing on cancers in dogs. So I was looking for opportunities to do more research work on canine cancers. And I was very fortunate to meet Dr. Jaime Modiano, who developed a cancer research program for dogs in the University of Minnesota Vet School. So I started my postdoc research fellowship in his lab, focusing on canine hemangiosarcoma by moving to Minnesota. So this was really my biggest move. I'm changing almost everything in my life in a good way. But I always feel happy doing my job for canine cancers. And at the end, at the University of Minnesota, I focused on cancer research in dogs. And I loved my job there. Although my family and I had to adjust to the totally new environments, new nature and people, but it was a really fun experience. And many of my friends complained about the cold weather in Minnesota, but we settled down very smoothly, and the weather was not really issue for me at all, because I really like my job, and that was my main focus. But realistically, in fact, my job was not very stable, because animal cancer research is a very small field and greatly depends on research funding status, and obtaining enough funding to sustain a research position focused on the cancer research, canine cancer research, is pretty tough. However, luckily and thankfully, I was very successful for 11 years in Minnesota. Then there was a really great opportunities offered by the UF University of Florida Vet School, allowing me to continue this work for canine cancer research. So I moved to Gainesville 2 years ago almost, and which is the palm tree and gator enriched instead of snow and gophers. Yeah. Then now I'm going to share what I have been doing for research. My research has been dedicated to hemangiosarcoma in dogs, starting in Minnesota. As many of you might know, hemangiosarcoma is really relatively common in dogs, but very rare in humans. Unfortunately, it is prevalent in certain breed dogs, such as golden retrievers. And it has been reported that about 60% of dogs, or 60% of all golden retrievers may die from cancers. And among them, 20% will get hemangiosarcoma. And survival time is generally very poor, with 3 to 4 months overall survival time, and only about 10% of dogs, or even smaller number of dogs, can live longer than 1 year. And standard therapy, including surgery and chemotherapy, can improve survival a little bit compared to doing nothing. Nonetheless, many dogs are euthanized in clinics at diagnosis due to the emergency status and aggressiveness of disease. So my research goal is to better understand why this cancer happens and why this cancer occurs and why it is so aggressive, and how can we change that. Again, my job is to make a change in cancer treatments for dogs. Then how can you do this? We need to do laboratory research to understand the basic biology of cancer, focusing on hemangiosarcoma specific biology. To understand this biology, we need to use the right technology. In general, it is very challenging to utilize cutting-edge technology for canine cancer due to a lack of resources, which are optimized and established for animal use and canine use. However, it is necessary to have for doing great science and good research. So we are working on maximizing the utility of cutting-edge technology for canine cancer research to have impactful outcome. And this slide shows the very first two meaningful research papers for hemangiosarcoma came out 20 years ago, back in 2004. One study developed an experimental method to produce hemangiosarcoma tumor tissues in mice. And this was a really significant breakthrough, because it accelerated research activities for canine hemangiosarcoma, and it is still used today. And almost simultaneously, another study showed the biological characteristics of hemangiosarcoma cells by growing them in the laboratory. They obtained the tumor tissues from dog patients, and successfully kept these tissues and cancer cells alive in the lab. And they froze cancer cells in a liquid nitrogen tank, allowing us to use them to this day. And Dr. Jaime Moriano, who was my previous research mentor and is now is a good friend, he started a program for studying cancer in dogs at the University of Minnesota. Before I moved to Minnesota, Jaime's team just finished the study using a method called microarray. And this technique allowed them to analyze 18,000 genes in the tissue, in the hemangiosarcoma tissue. Although it was very powerful, at that time, this method was old and almost stopped being used because much better technology came out, which called next generation sequencing. So when I started working with him, when I joined in his team in 2011, we quickly started using a new method to study hemangiosarcoma in dogs. And this allowed us to learn important clinical aspects for treating hemangiosarcoma because we found that some dogs might live longer than others based on our gene expression profiling data from this study published in 2014. And we also collaborated with other researchers from Harvard, MIT, and North Carolina and other places. Together, we found out which genomic place might make golden retrievers more likely to get hemangiosarcoma, published in 2015. So based on these findings, we extended our efforts to find cancer mutations that are more functionally important because these mutations can be potential therapeutic targets. The major finding of these two studies in this slide were cancer genes and cancer proteins that increase the specific cancer-related signals in hemangiosarcoma cells. There are specific chemotherapy drugs that are not currently used in dogs. So from these studies, we found that some hemangiosarcoma have a unique gene mutations that could be blocked by this drug. And these findings were from actual tumor tissue samples obtained from dogs after surgery, as well as from laboratory experiments. So we are planning to design clinical trials in dogs to test these new chemotherapy drugs based on our studies. I will get into more details later in this presentation. Then a few years ago, 2021, we applied another new techniques involving AI and machine learning. Specifically, we analyzed the large data sets of genomic profiles generated from canine hemangiosarcomas, as previously shown. We developed a new analytic computational method using AI and machine learning techniques. This led us to discover new gene signatures that could improve the diagnostic accuracy of canine hemangiosarcoma. I'm talking about the technologies, but hemangiosarcoma is a very challenging cancer to study and understand, and it is difficult to determine why this cancer occurs, how it grows, how it metastasizes, and why it is not easily cured. Technology has been advanced very rapidly these days. However, the advancement of cancer treatment has not progressed as quickly. So when we always apply new technologies for our field, for hemangiosarcoma, we always focus on finding something truly clinically important for hemangiosarcomas and sick animals. And we believe that our hard work and tireless efforts in identifying the key new molecules that regulate hemangiosarcoma specific progression, disease progression, will lead to the development of new effective treatments that can save more dogs' lives. And as the last publication of previous studies, I'm excited to share this paper, which has been recently accepted and will be published soon. This study is the result of a significant amount of work to further understand hemangiosarcoma biology for a long period of time, which was done by collaborating with many researchers, as you can see from this author list. And I'm proud to present this collaborative work. And I wanted to emphasize that our efforts are not those of a just single unit, but rather a multidisciplinary study. So from this work, we found that hemangiosarcoma has a very unique biological feature that creates a highly organized vessel controlling system. Hemangiosarcoma is a vascular cancer that destroys normal vascular structures, which can cause tumor rupture, especially splenic rupture, and results in emergency situation and sudden death. And so from this work, our finding delineates how hemangiosarcoma cells lead to vascular destruction, possibly causing hemorrhage in the tumor tissues. And furthermore, this information provides insights into the immunology of hemangiosarcoma tumor. Immunotherapy is an important area in cancer research. However, immunology in canine hemangiosarcoma is very poorly understood. So our study will help develop further research for immunology and for potential therapeutic approaches using immunotherapies for this cancer. And now I'm moving to what we are currently doing. And now my lab is working on multiple projects for canine hemangiosarcoma using different types of sophisticated technologies. As I mentioned, we always care about the potential for clinical applications and therapeutic developments from our research. So I will cover to that. And one of the technologies we are using is CRISPR screening. CRISPR is known as a genetic seizures, and we can change or correct the genes using this technique. In addition to the gene modification using this technique, this enables us to screen up to 18,000 canine genes at once and identify functionally essential genes for hemangiosarcoma disease progression. And we are also developing cancer models for hemangiosarcoma using induced pluripotent stem cells. And in addition to this project, another main focus of our work is developing AI models that can detect hemangiosarcoma cells in clinical images. The gold standard for colon cancer diagnosis is histological image analysis. The images are typically stained with a red and purple color, as you can see in these images and these images as representative. In routine diagnostics, pathologists evaluate these images, especially focusing on the shape of cells and tissue architecture in these images, and provide a confirmed diagnosis. However, hemangiosarcoma diagnosis might be challenging sometimes because they are very bloody and dirty. And in a preliminary, our preliminary study, our AI models successfully detected hemangiosarcoma cells in this type of images, highlighting the relevant areas, especially if you look at these blue-ish images. This model highlights the relevant areas in red and yellow colors, indicating the tumor cells. So the AI model can detect where the tumor cells are within these images. So in addition to this, using this AI model, we are also trying to find evidence in these images that provided us with clinically relevant information. In other words, we are developing AI models to predict whether dogs can live longer or shorter based on this kind of image features. Then now let me take you back to 2004. 20 years ago, we were able to visualize how the hemangiosarcoma cells looked. However, we were not able to understand the genomic and molecular features inside individual cells. For example, if you look at the SBHSA images here, let me see. You can see the individual cells indicated by red circles. like this, but you don't know what is inside them. But today, next generation sequencing and AI technology allows us to quantify the gene expression levels of nearly 18,000 genes per cell. It means that we can now understand what is inside individual cells. The figure on the right side shows the gene expression patterns from approximately 20,000 hemangiosarcoma cells, and each small dot indicates a single cell. And we have quantified the data of all 18,000 genes per cell. And also this figure shows the gene expression change between the cells treated with chemotherapy, drugs, and those without treatments. This way allows us to identify unique single cell populations that are resistant to chemotherapy. However, this cell identification is not that simple. It requires diligent work to prove, as I showed here, although I'm not going into the details of this data. But our effort was not very simple. So we did a lot of work for proving our finding biologically. And in the last two slides, I will show you how we plan to develop the new therapies for hemangiosarcoma. In our previous genomic study, as I showed, we found that 30% of dogs with hemangiosarcomas have specific mutations called PIK3CA genes. These mutations can be observed in 30% of dogs with hemangiosarcomas, and these mutations also detected in human cancers, especially human breast cancers. And there is an FDA-approved drug for human cancer use called Piqray. However, this has not been studied in dogs with hemangiosarcoma, although PIK3CA mutations can be detected in these tumors. So as I showed in the previous slide, we characterized hemangiosarcoma cells having the PIK3CA mutations to determine if they respond to this drug or not in our lab. So as a next step, we are seeking opportunities to test this drug in dogs with hemangiosarcoma from a dog clinical trial. But in the meantime, there are 70% of dogs having no alternative therapeutic options. So we will also continue studying other aspects of hemangiosarcomas in this case by better understanding of biology for these cases. And although our discovery of specific gene mutations is remarkable, developing new cancer therapy is not straightforward, because every cancer patient, every dog cancer patient is different. So a personalized approach is needed for effective treatments and proper disease management, clinical management. For doing so, we are collecting complex data sets from diagnostics lab, molecular labs, and clinics to build by collecting this complex data set to build an AI model that can predict clinical outcomes and drug response by sorting out the most significant fit molecular and clinical features and analyzing big data. So this is what I prepare today, and here is my very simple form of acknowledgement for this work, including my current lab members and the current collaborators at the University of Florida Vet School, as well as the University of Florida, University of Minnesota. For today's talk, during the short period of time, I have very briefly introduced our intensive research work done during a long time of period. So if you need more information, please feel free to reach out to me. And thank you very much for listening, and I'm happy to take questions.
I have a question.
Yes.
Pat,
It's
go ahead.
it's Pat. How can we help you at the University of Florida in the clinical trials that are coming up? Or I know that they have the hemangio vaccine, which is not your study, but what types of things can we as golden owners, when our dogs are diagnosed, if we want to help you with your clinical trials, or we want to do that, what is it that we can do? How do we reach out to you? What do you need? What do you guys require? Is my question.
That's a great question. And I'm really excited to hear about that, because we are kind of early phase, but almost a preclinical, pre-veterinary clinical studies are done. So we are ready for the clinical trials. But I just came here. I have been here for 2 years, 2 years, and I just started. And because most of my time here spent setting up a brand new lab and almost set up, and now normally working and operating. And then I'm not doing the clinic, but I'm in the Department of Small Animal Clinical Sciences, working with all other clinical faculties here. So I already had a discussion with clinical faculties here about these clinical trials. So because this drug has been approved for human use, it means that there was already approved toxicology study done in experimental beagle dogs for human use. So we expect that this drug is pretty safe, but we haven't tested this drug in actual dog patients. But what I'm saying is that we are ready. We need to do some paperwork and working closely with clinicians who is also interested in this clinical trial. His name is Maria Takeda, who is a radiation oncologist, and we also have other internal medicine oncologists as well. So for action items, we need to more closely plan to conducting these clinical studies. But to answer to your question, how you can help is that your interest from your groups and all of these people, and appreciation to our work and support, like emotional support and other supports, it will be great for motivating us to move forward.
If there is a time in the future where you do need dogs or whatever else, I assume then you would reach out to us, our club, if you were looking for animals or anything like that. Would that be helpful?
Yeah, that would be great. Yeah, because especially if dogs from in local place, that will be much probably easier for managing. Although we could do the dogs from other states. All right.
Did I recently read that there's a PIK3CA test?
Can you say again, because your internet quality was not.
Sorry. I think I recently read that there's a PIK3CA diagnostic test. Is that
Yeah,
something you have to have a tumor to stick, or is it a blood test?
This is actually we need a actual tumor tissues for
Okay,
testing.
so it's not just okay.
So ideally, if we set up this trial, we need to have dogs most likely in spleen, allowing splenectomy, the surgery. So, and then we can get some small piece of tumor tissues and do genetic test, which is much more accurate than the blood testing. because we might as well obtain the tumor tissue by surgery, and that would be an ideal case. Although we are working on the PIK3CA mutation discovery from the blood test, but as far as I know, there is no highly accurate blood testing for detecting these mutations from the blood.
Thank you.
So John, if a dog were to become sick with hemangio and they were to have their spleen taken out, which I certainly have had two that have, are you in a position to accept from vets, if they can ship them to you, those spleen tumors, or are you not in a place that you need those yet? Or if we have dogs that are diagnosed and are going to have their organs taken out or the tumors taken out, is that something you'd be interested in knowing or getting from vets or whatever for any of our dogs that might have hemangio and end up having their tumors extracted?
Yeah, that'd be great. What I have done in Minnesota was that the best thing is to get the actual tumor tissue right after the surgery or within a day directly from the clinics, the university hospital. But we also got delivery of the tumor tissue samples in a specific liquid media by overnight shipping. Sometimes when they from other states, local clinics send the tumor tissue, some of the piece of the tumor tissues by shipping. On Friday, it took like 3 days when I got the tissue samples on a Monday. But still, the 3 days would be within the 3 days, the tumor cells still are viable. So we still were able to use the tumor samples, at least for, especially for DNA testing, genomic testing is much safer to have the 3 days long tissue sample. So again, it'll be great to have the actual tumor tissues. And if we know in advance when the dogs get surgery and it's a plan to get surgery, and we are going to be ready for getting that tissue samples and do the following experiment.
So I have a question. So if people, and a lot of people in this Central Florida area, if their dog gets cancer or is sick with these things, they end up going to UF anyways, because that's kind of our, our gold standard vet school around here. So do you guys have a connection where if those dogs come into that clinic, that you guys get those samples and can do research from that? Are you working with them directly, or is there a contact also that we can use, like that we can put out to our club members, like Pat was asking, if your dog is sick and is elsewhere in a different vet clinic, that we could maybe connect them so that they would know if they want to participate in sending a sample?
Yeah, again, unfortunately, to be honest, I'm not doing clinic. I don't know what's the realistic and actual situation between the animal hospital and the local clinics here. But I believe that I believe that there is a strong connections between the UF Animal Hospital and the local clinics, because the Dr. Rowan Miller, who has been here for over 20 years as a veterinary medical oncologist for the vaccine trial, he has been doing a lot of clinical trials. So he knows much better than me for sure about that. But I don't have any the negative or impossible ways to work with closely work with the local clinic.
All right. Go ahead, Rob.
Yes.
Is that liquid, the medium you talked about, that it has to come to you in? Is that something that most veterinary practices have on hand?
Oh, can you say again, please?
Medium
Your
for the liquid.
your internet was.
I heard. I'll say. She was wondering if the medium that they need to send the samples in, the liquid, if that's something that
Okay.
clinics have.
No, actually, they don't have. So, what we did before when I was in Minnesota is that we just distributed our medium to the clinics who are interested.
Ah,
So
okay.
we sent like in the 50 mil tube like this,
Yep.
and then we distributed these tubes containing the special media for
Okay.
them to have.
Okay,
Mhm.
thank
Or even it is still possible to use the routinely use the media the just PBS, just normal saline
Mhm.
um used in clinics. That is that also works if they don't have a specific, especially if can be delivered in a day or by just overnight
Yeah.
shipping. It is possible highly.
That's good to know.
that is good to know.
Mhm.
It's Pat again. I just for the club's interest, that's why I asked the question. There is a club member from the Everglades Club who found that her dog had hemangio. And we got very lucky that Dr. Kim, we happened to be talking to him about something else. And on the day of the surgery, she found out that she could, in fact, send that tumor tissue to Dr. Kim. And she's in Miami. I believe that was right. I don't think she was up at UF when they did the surgery. Maybe she was, but they were able to, just as a vet that wasn't associated with UF, they were happy to do what they could do, and they did send the tumor tissues. So it kind of depends on your vet too, whether they're willing to, help out and try and get it there. Even if there's no relationship, which there wasn't there, they were able to get those tissues to Dr. Kim. Literally, they were still they were going to throw them away or get rid of them, and she was able to call back once she did that and say, "Can you get them?" And they actually pulled them out and did and sent them. So your vet can figure out a way to do it if they work with you.
Great.
Yeah, sounds good. Yeah, because we haven't set up this task which is more like systemic. So it'll be good to have some like paperwork and then make some guidelines and
Mm.
milestone and action items and this description for doing this, and having more close discussion with the clinical faculties here, and possibly making some like sheets or the brochures informing our plan and our method to other clinics to have, because everyone is busy and clinical faculties are busy with high caseload. So although there is interest by others, but I need to more actively and closely working with them to be prepared more properly.
Great.
I feel sure if you'll come up with those guidelines that every one of us will give the sheet to our veterinarians.
Yeah, we've got a lot of golden owners here in Florida that are very invested in these studies. So whatever
Yeah, it's
help
really,
you lend.
If nobody else
I'm really
if nobody else has a question, I just want to finish by I just want to say one more thing. I think clinical trials are one of the greatest things that people can do when you love your dogs. I have, Mike and I have been in probably 4 or 5 of them with Dr. Modiano over the years and with other doctors. And, there's nothing that makes you in a terrible time feel better than to realize that there's something you can do that won't change the outcome of what your dog has to go through or what they do, but that you can take something and that your dog is giving to the future of saving other dogs and is making a difference. I am proud of all of my dogs that have been through clinical trials. And I'm proud to say that it made me feel better that in losing them, that I felt that they were doing something good for the dog community and the breed. So I'm a huge encourager of, if you find yourself in a situation where your dog fits into a clinical trial, especially one that's this close, because that doesn't usually happen, but one that's this close by. I encourage everybody, if you unfortunately find yourself in that position and situation, that you think about reaching out. And, it's just a great feeling to know that, you and your dog did something for everybody else. So I appreciate your work, Dr. Kim, and I wish you good luck with it.
I really appreciate these opportunities.
Yeah. Thank you so much for taking your time to come and talk with us this evening.
No problem. It's really honor, and as I mentioned earlier in the introduction, this is very excited because I cannot imagine having this meeting if I were just stay in my home country, and before I have this great work for hemangiosarcoma. And I love golden retrievers. I love all the animals, although I don't have one yet. But so I really enjoy it. And I mean, it is kind of dilemma because this is devastating dogs the disease. It is hard to manage and it is very sad. But it is what it is, and I'm trying to do make something change.
Thank you. We so appreciate your work.
All right, thank you very much again. And so yeah, just feel free to reach out to me if you have any questions or interest, and I'm here, and I'm always checking the emails. So
Wonderful.
all right.
Are you okay if we send out your clinic information or your contact information there to just our club members in case something comes up where they need to connect with you about any research?
Sure. You can share my order information.
Wonderful.
That's good.
All right.
Thank you very much. Should I leave the meeting?
Oh
You
yeah,
stay
Cindy,
on.
All right.
Cindy needs to make herself the the host again. You're muted, Cindy.
I think I just
Thank you,
made
everyone.
co-host,
See you
but
later.
let me double check.
Thank you.
Yeah, he better not leave for all of us.